This is a video about meiosis by Ansia,
Aaliyah,
and Taisha.
What is meiosis? Meiosis is the division of sex cells.
What are the stages of meiosis? Interphase. This phase begins with the plasma membrane of a cell. Below the surface of a plasma membrane lies the cell's nucleus. Across from the nucleus are two centrosomes, which are connected in pairs. So I kind of understand what's on this paper, but not all the way. So what does this plastic bag represent? The plastic bag represents the nuclear membrane. So what is these yellow and red beads supposed to represent? Well, eventually they'll form into the chromosomes, but right now they're the chromatins. Okay. So what is this white piece right here? That is the centrosome. And what are these? Those are the centrioles. Okay. So that's what anaphase looks like.
The first stage in meiosis is prophase 1. During prophase 1, DNA coils tightly and the homologous pairs of chromosomes become closer and closer due to synapses, and the chromosomes cross over and exchange segments.
Right to my left is a model of prophase 1 during meiosis. As you can see, there are two centrioles on each side of the chromosomes, and here is an example of the chromosomes coiled tightly together. And these are the centromeres, and that's prophase 1.
The second stage in meiosis is metaphase 1. During metaphase 1, the nuclear membrane disappears. The microtubules create a spindle fiber. Those spindle fibers attach to one side of the centromere. The homologous chromosomes stick to the microtubules from opposite sides, and each homologous chromosome line up at the metaphase plate.
As in the previous phase, we have two centrioles on each side of the chromosomes, and we have the spindle fibers. These are strings representing the spindle fibers that the macrotubules created, and they're connected to each centromere, beginning the process of them separating. Ends up with the complete haploid set of chromosomes, which makes up one number of the homologous pair. So now this is the process of anaphase 1. This is the chromatids that pull in towards the microtubules, the centrioles. This right here is the centromeres. These are the chromosomes, and they have two different colors. They're mixed, have a combination because they did a crossing over. And when they cross over, they mix the genetic genes up, and the genes are located at the tips of each chromosome. And that is anaphase 1. The two sister chromosomes will not be the same. The cells rest at endokinesis before entering the second stage of meiosis. In cytokinesis 1, simply clear, the two daughter cells are produced, which create diploid cells. Taisha will explain to you now what is really going on in the final product of telophase and cytokinesis 1. Well, here we have two diploid cells, and it has transformed from two separate chromosomes to chromatin, and we have the what are these called again, Annie?
Centrioles?
Centrioles. We have the two centrioles beside each cell because they're now two separate cells, and they are not identical because of the crossing over it has made throughout the entire process.